Hello dear students, let's start with the first lecture of this new session with our very favorite subject medicinal chemistry second. Its pharmacy code is BP501T. T stands for theory. The first topic is histamine and antihistaminic drugs. So in today's lecture we will go for the introduction of histamine and the types of histamine receptor. So histamine is a nitrogenous neurotransmitter. Nitrogenous means it is made up of nitrogen, and it is a neurotransmitter means it transmits the signals from one place to another place. The histamine is involved in local immune responses like allergies and hyperacidity. The allergic conditions which are caused by increased production of histamine include number 1, rhinitis, which is called as inflammation of nose. The rhinitis is also called as hay fever. The allergic condition also include the rhinitis conditions like sneezing, runny nose, red, itchy and watery eyes, dermatitis, that is inflammation of dermis, asthma, that is shortening of breath. Let's look at the chemical structure of histamine. If we look at the chemical structure, we find that here is the presence of nitrogen in amino group and inside the heterocyclic nucleus. In this case, the heterocyclic nucleus present is imidazole. For IUPAC nomenclature, we will select this 2 carbon chain. So this carbon is given number 1 because it is attached with the amino group, and adjacent to this is carbon number 2. So in IUPAC name we call it ethane 1 amine. We call it ethane because it is made up of 2 carbon. 1 amine stands for the attachment of this amino group on position number 1. So it is ethane 1 amine. Now on carbon number 2, we have presence of this imidazole ring. For imidazole, the numbering start with this nitrogen which bears this nascent hydrogen. So this will be number 1, 2, 3, 4 and this will be number 5. So we find that the imidazole ring is attached with its position number 4 on the second carbon. So we say it on to 1H imidazole 4-il, followed by the name of the basic moiety ethane 1 amine. So the systematic IUPAC name is 2, that means on this 2, 1H imidazole 4-il ethane 1 amine. Next, look at the biosynthesis of histamine. The biosynthesis of histamine takes place in Golgi apparatus, which is also called as Golgi complex, and this is present in most of the cells. Of our interest is the mast cell which are present in skin and other areas. The basophils, one of the type of white blood cell, neurons in the brain, and the parietal cells. So the biosynthesis of histamine takes place in Golgi apparatus of the cells. The starting material for the biosynthesis is this amino acid, which is called as histidine. It is an alpha amino acid. This is the carboxylic group and this is the alpha carbon. On alpha carbon we have the amino group, so it is the example of alpha amino acid. So the starting material is histidine. This histidine is acted upon by the enzyme L-histidine decarboxylase. Now look at this name very carefully. D means removal, carboxyl means removal of carbon dioxide. So whenever this enzyme acts on histidine, there is liberation of this carbon dioxide from here, and this whole liberation takes place in presence of pyridine phosphate. So with the release of carbon dioxide, we get the formation of histamine. So just to look at the starting material and the final product. There is the difference of carbon dioxide only. Next, move on to the storage of histamine. The storage takes place in the secretory granules of the mast cell, basophils, and the parietal cells. These secretory granules are also present inside the Golgi apparatus. Now let's look how histamine is released during its action. So whenever a foreign particle, which we will call allergen, that means which can produce the allergy. So whenever a foreign particle attach on the cells like the mast cell, basophils and the parietal cells, the attachment takes place on the immunoglobulin E. That is a protein which is found on outer layer of the cells. So whenever any foreign particle attach itself onto immunoglobulin E, so there is cell activation, and this cell activation brings the contraction of the secretory granules, due to which whatever histamine is present in the secretory granules is get released out, and that is that released histamine which produce the allergic reactions. Now let's move on to the types of histamine receptors. There are 4 types of histamine receptor: H1, H2, H3 and H4. The number 1 is H1 receptor. This H1 receptor is located on the mast cell of skin, the bronchial smooth muscles and urinary tract. Whenever histamine attach on H1 receptor, it gives some actions, and these actions include vasodilation, that is dilation of the blood vessel, and due to which there is increased visceral formation leading to rhinitis. So here is a picture of vasodilation. Here you can view that this first picture is of the normal blood vessel, while this is the dilated blood vessel. So whenever histamine bind on H1 receptor, it gives formation of vasodilation. The other action includes dermatitis, that is inflammation of the dermis. Next action is asthma, that is bronchoconstriction. which ultimately leads to shortness of the breath. Next action is urinary bladder contraction. Though this is observed in very few cases, the contraction of the urinary bladder will lead to urgent urination. That means the person who undergoes this contraction has to urgently go for urination. The next and the last action is urticaria, that is skin rashes with red patches on the skin. Now when histamine is attached on H2 receptor, it leads to hyperacidity. The H2 receptors are present on parietal cells of gastrointestinal tract. These parietal cells are involved in production of hydrochloric acid in GIT. So whenever histamine bind with H2 receptor, the parietal cell undergoes contraction and there is increased release of hydrochloric acid. So this increased release of hydrochloric acid leads to hyperacidity. The next type is H3 receptor, which are located in the central nervous system, that means inside the brain. So attachment of histamine on H3 receptor gives a negative control. Negative control means there is decrease in the release of histamine itself, and beside this there is decrease in release of acetylcholine, norepinephrine and serotonin, which are the other types of neurotransmitters. The last type of histamine receptor is H4 receptor, which is present on the basophils, that is the type of white blood cells. The action produced is chemotaxis, that is the movement of foreign particles including the bacteria from low concentration to the high concentration. So the H3 and H4 agonist and antagonist are not included into the syllabus. We have to largely focus on the H1 and the H2 antagonist only. Now the therapeutic application of H1 and H2 antagonist, the first is the application of H1 antagonist. Now one very important point is that the H1 antagonists are also called as inverse agonist, and they are used to treat rhinitis. The rhinitis is inflammation of the nose, and it include some allergic conditions including sneezing, the redness of nose, the red, itchy and watery eyes and runny nose. Dear students, you may think that this kind of condition we can observe in most of the allergic conditions, and this observed during the change in the season. That means whenever we enter from summer to the monsoon season, we get these allergic conditions. The next is treatment of urticaria. Now dear students, if you look at this picture, here is the red patches development on the skin. So this condition is called as urticaria. The H1 antagonists are also used in treatment of itching. many a times most of the peoples are get affected by such allergic conditions. The H1 antagonists are also used in treatment of asthma, that is shortness of the breath. The H2 antagonists are exclusively used for treatment of hyperacidity, that is the increased production of hydrochloric acid inside the body, inside the stomach, due to which there is chances of formation of ulcers on the intestinal linings. So H2 antagonists are used for treatment of hyperacidity. Hyperacidity in most of the cases is caused by consumption of fried foods. So these are some pictures of hyperacidity. So H1 and H2 antagonists are used for treatment of allergy and the hyperacidity conditions. So that is all about our first lecture. We will go through the classification of the antihistaminic drugs into the next video. Thanks for watching. Please like, share. Thank you.